Ever wondered what it's like inside a university biology lab? Same. So naturally, U of I sent me, a music major, to give you a tour.
Like your bangs, tuck those back and then put the wig over it.
Okay.
I can see it right here.
Okay, what am I looking at? What am I looking at? Okay. Oh my gosh. Hi, I'm Rainey. I'm the student social media vlogger for University of Idaho. I'm not a scientist. I have absolutely zero experience in a lab. Still, back in December with a different haircut. I toured one of U of I's Biosafety Level 2 labs, where students safely work with organisms that could be potentially harmful if handled the wrong way. The lab is led by Associate Professor Paul Rowley, who asked 2 students to give us a tour. I didn't go alone. I brought a videographer with me. Say hi, Garrett.
Hello.
Instead of just giving us a tour, the students gave me a challenge. I had to complete something using the tools in the lab. They also asked me to bring 2 things. One to test the dress code, and the other, a flower. You'll see why later. It was a ton of fun. Here's how it went. It is snowing out and I have never been to this building. I've only seen it walking to class, and I am probably gonna have smudges on my glasses for the entire time that I'm in there. So I honestly have no clue what I'm getting into. I don't know what a level 2 lab means.
Maybe it's how many stories the lab has. It's a 2-story lab.
It's a 2-story lab?
No, I just made that up.
Okay. They have a blue roof and there's pine cones making a shape on them.
Let's make our way down there.
Okay. I don't know what room it was, 156 through 157. Rowley Lab is this.
This is the Rowley Lab. What are we doing?
We're gonna see if they'll let me in with excessively long hair, which I currently do not have, hence the purchase of a very nice wig.
Wow. Oh, you look so good.
Okay, good.
It looks really natural. Tuck your hair back though, like your bangs, tuck those back and then put the wig over it.
Okay.
Wait, post up by the Rowley Lab thing. Give me some kind of pose with it.
We're at the Rowley Lab.
Look like a person, like somebody's
I
looking
think
up.
it is a person. Do you think that's supposed to be Rowley?
It's Rowley.
Rowley.
Rowley? Rowley.
Let's
Oh, we've
figure
been saying
that
it
out.
wrong. Okay, is that him? Is that what he looks like?
Yes, exactly.
That's exactly what he looks like?
Yeah.
Okay, great. They have Totoro. Is it Ghibli or Ghibli?
Yeah.
Okay. I don't know what to do, so we're knocking.
Hello.
Hello, I'm Rainey, and I'm told that I'm getting a lab tour today.
Yeah, you are getting a lab tour.
I have a rose for you. I don't know what it's for.
We'll use it later.
We'll use it later. Also, do I need to tie up my hair, or is
Probably
this?
should tie up your hair. No,
Oh, okay. Then I don't need this.
Well, welcome into the lab.
Wait, who are we talking to?
Sorry. All right. So I'm Rainey. Who's giving me the lab tour today?
Me, Jack, and
I'm Lily.
Okay.
I'm an undergraduate.
Undergraduate. And then is your major biology, or is that?
Major's microbiology.
Microbiology. Does that mean there's macrobiology, or just that?
No.
Okay, sorry. I'm a business major, so I'm in microeconomics, and I was like, maybe it's the same. Nope, not at all.
Nope.
Are you undergrad?
I'm a master's student. Actually.
Master's student?
Getting my master's in biology, regular.
Biology. Okay, not the small one.
Nope. Just the
Okay.
regular one.
And so where are we going?
You can just come right in here. We'll show you around and we can come back and get you a lab coat.
Oh,
for
okay.
Activity later.
Wardrobe
This
change.
is where lab coats are and stuff. These are cell culture.
Oh.
Brooklyn has human cell line growing
Interesting.
in there that she uses for her work.
Caution, use of toxic, explosive, or flammable substances in this cabinet should be evaluated by your appropriate safety personnel.
Oh, my
Oh.
ears.
Okay.
Well, I just figured you weren't gonna be able to hear anything we were saying.
No, that was great.
So if those make it a level 2 lab, does that mean there's level 1 and 3, or is?
Yeah. So
Okay.
Biosafety Level 1 is work that is done on like a bench top, just
Okay.
by like a flame, with like non-pathogenic
So it's
organisms.
just like your Bunsen burner stuff? Okay.
Yeah. And then we do Biosafety Level 2 stuff in this lab. So we use do work on
Okay.
opportunistic pathogens. So this is a yeast. This is Candida glabrata probably. where the stuff grew is where the when you streak it out with like a stick, where your stick touched is where stuff grows. So and we want to get ones that are just single colonies over here, so you can easily just pick from single colonies instead of the region that's like thicker. When we work with these, it has to be done in our biosafety cabinet, which has like special ventilation, so it circulates the air through repeatedly and it filters it, so it keeps all the pathogenic stuff in
one space.
In one space. It keeps you safe from the work.
Is this like for the purposes of like medicine or?
No. So some of our work that we do can have potential medicinal implications, but a lot of what this lab is about is working with yeast, and specifically yeast that produce killer toxins. And so killer yeast make killer toxins, and these killer toxins kill other yeast, basically. And so she talked about Candida glabrata, which is a human pathogen. But like some of the work that we do kind of investigates, could we use killer toxins to kill human pathogens as a potential idea for a therapeutic? Having research that investigates these types of relationships is pretty interesting, and could really benefit a lot of people in the future.
So I am learning about how they study pathogens inside of yeast cultures. Like, is that how you'd say that? Or
yeah,
like,
sure.
Okay, sure.
There's another little interesting piece of equipment that we use. So this is our negative 80-degree freezer.
And then we can like look and we can pull out.
And you have to wear the special gloves?
Yeah, because you'll freezer burn your hands. And then we have like boxes everywhere in.
Oh my God. Now we're on to the lab challenge, where Lily and Jack had me practice Jello.
See, what size lab coat do you think you wear?
Very large.
Yeah, real large.
And why were we not wearing lab coats before?
Well, you don't really, if you're not doing any work like on your bench top or, touching organisms, then there's not really a need for it.
Wait, I have an idea for a transition. Boom! Look at that. Instantly on.
Do I need to button it?
You should button it, yes.
Button it all the way.
These are so small.
Sweet.
Oh my gosh. Are these latex?
No, they're not.
Okay.
Are you allergic to latex?
Yes, I am.
Oh, wow. You just put those on without even asking.
I'm so ready for whatever I'm going to be doing.
Nice.
So we are going to run a load and run a gel. So I have some samples I have. So these are plasmids. Which are little circular bits of DNA. We want to just confirm that what I think is in here is in here. So DNA is negatively charged, and so we're going to put it in this little guy. And it's going to run towards the positive charge, and we'll be able to see like how big my DNA is, if it's there at all. So if you look right here, like look above, you can see there's little holes in there. And so what we're gonna try and do is we're gonna put some of this liquid into the little holes. In the gel. This is ladder. So it looks like this. So it's just like a template, I guess, to show how big different pieces of DNA are. So we have something to compare our sample to. I will prepare the sample for you, and then you can just try and pipette it into the well. And you'll probably miss it entirely.
Great. How long did it take you to get good at this?
I don't know, a couple months.
Months?
It's hard.
I believe in you though, you got
it. I've got it.
You try it a couple times. So I'm gonna mix this with some orange loading dye so that we can see it. So you can see
that little dot, that is what we're working with.
So we have a pipette right here, right? And so a pipette sucks liquid up. And so what you're gonna wanna do is you're gonna push it down to the first stop, and like you can clearly feel the resistance. And then you're gonna put it into the liquid, and then you're gonna release it, and that'll suck it up. And then you're gonna press it down
to get it in there. And I'm doing that?
Put the little tip into the liquid and then release it. So now it's in the pipette. You did good there. And then you're just gonna really slowly try and put it into that next well. I actually think most of it made it in. Yeah, you actually, I think, got most of it.
Most of it?
Yeah. So then I will load a sample, and then we can run this, and we can see later if it actually
works. Do I need to change my major?
Yeah, sure.
Are these people being labeled as absorbent pads, or is that just what's in the cabinet?
These are all alumni. So it's all people who have worked here, who have graduated and stuff. And so now you can take this, right? And this is our positive charge here, and this is our negative charge. So it's like a big magnet. Okay. And the DNA is negatively charged, so it's going to go towards the positive charge. Okay. And so we're gonna leave this for like 40, or not, so we'll run it for like 20, but we'll be able to see where the DNA goes. And so then we can leave that and we can go do other stuff. Okay. We'll come back and see if this works.
Cool. All right. So I can take my gloves off.
Yeah, and so then you want to put your gloves in the biohazard waste.
Did I touch a biohazard?
Usually, not right now, but we usually use ethidium bromide for our gels. Okay. Which is a carcinogen, so that's why.
Okay, yeah.
But we did not use that.
We did not use it. Okay. And I did the part where I would have used it before you got here.
Okay.
All right, so this is the first room of our lab. There's 3 total. This is a lot of where we do gel loading and gel analyzing. There's some interesting stuff we keep in the freezer, just a bunch of DNA basically. Here's our incubators that we use to culture our yeast so they can grow up nice and strong so we can test them. Then right here, a centrifuge. Okay. These are littered around our lab, but this is a nice small one. There's an even bigger one over there. Yeah, this is a tabletop centrifuge. This one can hold larger volumes. You can fit all these tubes. And then we have 2 more incubators flanking either side, so more culturing at different temperatures. Okay.
And then this is our laminar flow hood. So we mainly pour plates in here, and so we make the goop that goes in the bottom of petri dishes. Okay. And you pour it when it's really hot, and then it solidifies, kind of like Jell-O. Yeah. And we do that in here primarily, or on a bench top by a burner. And then this is where we make all our media and reagents and things. And all of our powders that we make stuff with are in here for the most part. And then maybe if you want to take a whiff of the yeast extract, it's got a real nice strong scent to it.
Oh, yep, yes it does. Oh, that's great. That smelled like bread that's been fermenting for too long. Don't know how else to put it. This is a very detailed biology chart. They have different species of different animals, and that. And then back to what this is.
This is a water bath. So it's just I don't know, it's like a crock pot.
Bath of water,
just has hot water in it. And you put stuff in it to let it cool slowly. Okay.
And who's your friend?
That's Julia. She is a previous undergraduate, but when she graduated, her parents gave her that cutout, and she left it here. Oh. So it's a Julia shrine, I guess. One of the last things in this room is our chemicals right here.
Oh, you have caffeine.
Yeah, that's what I was going to try and find. Where is it?
It's right there.
It's right here. We have 100 grams of caffeine. Okay. So I think a very small amount of this would kill you if ingested. Yeah, so what we had you bring the rose for is this is our liquid nitrogen. So it's way colder than our -80. And so we keep tissue culture, cell lines in here, human cell lines, stuff like that. So not yeast and bacteria. Yeast and bacteria are fine in the -80.
Yep, you got it.
And so you can see it's
oh my gosh, it looks like it's on fire.
Now you can just
and then
toss it against the ground.
I'll give you a glove and you can toss it against the ground if you want. Yeah.
Okay.
Yeah.
Oh, it did shatter. Oh my gosh. It sounded like Legos.
Yeah.
So why did we let Dr. Derp kill our flower?
So, I don't even know. I don't even know who Dr. Derp is or where they came from, but they've been here as long as I've been in the lab.
Do you use the same coat for all 4 years?
I mean, yeah. I've gotten a bunch of free ones. Okay. Embry, which is the program that has paid me both summers I worked here, has given me one each year. And if you look at this, you can see that the colors have moved further down.
Yeah.
Take this guy out, put it in this guy. This is our gel dock. Put it in the middle here. This will image our gel for us using UV light. It's imaging it.
All right, so I think this imaging is gonna tell me whether or not I failed the lab challenge.
This is our ladder. So we can see the different like sizes of DNA. There's files on this probably of successful ones. I don't know why the ladder's in the middle.
So this is a successful one, and that would be the one that we did. Gotcha.
All right, Lily, how did I do on my lab challenge?
You did pretty good. I think you actually got the stuff in the well, which is impressive for your first time using a pipette.
Okay, so if you had to grade me on the lab challenge, what would I get?
I don't know. A minus.
A minus? Okay.
And
I'll take it.
Let's see, this guy's little jiggly thing.
How does your experiences inside of one of these labs compare to your experiences inside of a biology lab that you guys had in high school?
It's not even close. I think the most complex thing I did in my high school biology class was make methane bubbles and set them on fire. So I've never worked with live organisms before until, coming here. So it's an invaluable experience.
If the lab is focused on research, how's that benefited you? Like, has that just made it so you feel more confident while applying to grad schools, or is that, has it built like your resume more, or?
Yeah, well, since I want to pursue a career in research, it's obviously like very directly related, but I've had the opportunity to contribute to 3 publications, and I've done 7 poster presentations and 2 talks at conferences, and then I've won like 5 grants. Okay. So there are lots of other things I contributed to beyond just being in here and doing lab work that I think have given me a good skill set that I can go on to use in my already future career.
Okay, and then you said you wanted to go into medicine. So has the lab helped prepare you in any sort of way for a medicinal field outside of understanding research?
Outside of understanding, yeah, to some extent, because, I kind of discovered that some of the research I'm doing could have medical implications down the road.
Okay.
But more than anything, I think it's good to have when you apply to medical school. I mean, the whole reason that I even wanted to do this master's was to get research for my medical school application. So, yeah, and it's been extremely vital to that. It gave me the experience to kind of talk in a more professional way, put together scientific data and present it to people. So yeah, it's a really good experience, and it's only helped me.
All right, thank you, Lily, and thank you, Jack, for the tour of your lab. I really enjoyed hearing about your research and seeing what you guys do here. Where is the exit?
Well, you can exit right down that way.
All right, do I keep going?
I think you keep going.
Okay.
Say bye.
Bye, thank you. Huge thank you to Jack and Lily for showing me around the biology lab. It was a ton of fun learning about a field that I don't really think I plan on studying in, but I had no clue that the research that they're doing goes on here at the University of Idaho. And anyways, I have to go to class. Like, subscribe, and as always, go Vandals.